It's a great pleasure to welcome Karissa Sanbonmatsu to Rutgers, virtually at least. I've known her for, for quite a while, but not in her original incarnation as an astrophysicist and plasma physicist. Only later, she got a PhD at University of Colorado at Boulder. Then after moving to Los Alamos after that, switched over to our field, structural biology and simulations. She's extremely well known for large scale simulations. I guess one of the first, or the first million atom simulation and the first billion atom simulation. Is an expert on the ribosome and other large structures, has done a simulation recently on genes. I'm always impressed that she was willing to jump into experimental work as well as computational work, and now has a major part of her lab doing both. There's also a very interesting TED Talk that you might want to search for on the web that I found very enlightening. Today, the title of her talk, I hope I have it roughly right, is about ribosomes and combining data from cryo-electron microscopy, single molecule FRET, and molecular simulation. So Karissa, please go ahead.
All right, thank you for the introduction, and thanks, David, for hosting me and having me here. It's really an honor to be speaking here. So I'm going to try to share my slides. Okay, can everyone see the title? Okay.
Yep, your slides look good.
Okay, great. So I'm going to talk to you today about some work we've been doing for, we started on this in the year 2000, so it's been a long time we've been working on ribosomes. We saw a lot of events like the X-ray structures of each individual subunit, and then the X-ray structures of the combined bacterial complex. And we saw the rise of high resolution cryo-EM this entire time, and then a lot of great single molecule FRET data to try to look at the actual mechanism. And in terms of ribosomes, we've mostly been doing computational work in terms of molecular dynamics simulations and some structural modeling. And we believe that MD simulation is a nice way to integrate all these different kinds of data, structural biology, biochemistry, mutational data, and single molecule FRET and so forth, to try to get a coherent picture of how the bacterial ribosome works. I should mention, with the high resolution cryo-EM, it's really opened the door to all kinds of studies on human ribosomes and mammalian ribosomes, a lot of cancer applications, and a lot of biomedically important applications of ribosome research. And then even with all this data in hand, we still don't know how the ribosome works. And so we continue to soldier on to try to figure it out. Okay, so I'm first going to give just a short overview of protein synthesis in bacteria. And then I'll talk about this kind of pipeline we set up over the years to use MD simulations to try to interpret cryo-EM and single molecule FRET. Then I'll talk a little bit about methodology. We have a new paper out looking at FRET dyes in proteins. And then I'll talk about some of the newer algorithms we have for fitting cryo-EM data. And then finally, how we apply those to try to understand these large-scale conformational changes of the ribosome. So what does the ribosome look like? This is a schematic of the 70S ribosome complex in bacteria. And we have two different views of the ribosome. The ribosome basically has two pieces, a small piece and a large piece. And then sandwiched in between is the genetic information, the messenger RNA that carries the information to make a protein. And then also these transfer RNAs that carry building blocks or amino acids to the ribosome. And then, so that's shown here on the left panel. On the left little cartoon in yellow is the small subunit. And in turquoise is the large subunit. And then in red is this messenger RNA that codes for the protein that's wrapped around the kind of neck of the small subunit. So the small subunit, we say, has a body, the lower part, and a smaller head in the upper part, and then a neck region that the mRNA wraps around. And then in pink is the tRNA that carries one amino acid at a time. And then it assembles this protein that goes through this exit channel in the large subunit. And then looking down on top, at first they thought the ribosome only had 2 binding sites for tRNA, the aminoacyl or the A site, and the peptidyl or the P site to accommodate 2 tRNAs inside the ribosome. And then Knut Nierhaus was instrumental in discovering E site, the exit site. So now there are 3 sites for tRNAs to reside in the ribosome. And essentially there's sort of the green tRNA is really where all the business is happening and the assembly of the growing polypeptide chain is happening. E site is where it's finished and it's ready to be shipped out into the solution. And then A site is where the tRNA is kind of on deck and it's ready to hand its amino acid into this growing polypeptide chain. And there are many stages, I would say about 20 or so substeps of this process, and they can be broken down into 3 major stages: initiation, where the complex assembles; elongation, where the protein is made; and then the whole thing disassembles and it's also recycled. So termination, recycling, where everything gets ready for the next protein synthesis event to happen. And then inside elongation, there's also 3 phases there. There's decoding, where the ribosome has to select the correct tRNA. Basically, it has to select the correct amino acid corresponding to the information on the mRNA. And so to do that, it has to select the correct tRNA. And that means it also has to reject incorrect tRNA. So this we call decoding or Trna selection. And then there's the chemical reaction, peptide bond formation, where that amino acid is added on to the growing protein, and that's where the chemistry happens. And then movement or translocation, where the tRNAs have to move by exactly, and the mRNA have to move exactly 3 nucleotides through the ribosome, and then that's a cyclic process that repeats. And in real bacteria, it requires 2 GTPases, elongation factor TU, that recruits the tRNAs to the ribosome, and then elongation factor G that helps facilitate movement of the tRNA through the ribosome. And then you have a lot of antibiotics. The ribosome is the target of about 50% of the antibiotics used in US hospitals today. It's a very critical component of all living things, especially bacteria. And these antibiotics here shown in green kind of act as little monkey wrenches that either grind the whole system to a halt, they can clog up that exit tunnel, or they can actually cause misreading errors and make malformed proteins. And actually in COVID today, there's a number of antibiotics used to treat a lot of the infections that people are suffering from when they're in the hospital nowadays. And then in terms of the Trna selection stage, this is just another cartoon illustrating this. And so basically you have the ribosome sitting there, and then what it's trying to do is pick the correct amino acids that's going to go into the protein that it's trying to build. And so it's looking through all the sea of tRNAs, 21 or many different tRNAs coming in, and then that's shown here kind of in detail, where the tRNA has this anticodon end to it, and then it has to match that to this 3-base codon in the messenger RNA that corresponds to a specific amino acid. And if that pairing is correct, then the ribosome detects that it's basically correct and then incorporates the amino acid. And we know that from experiments before the 90s even, where they would do mutations on the ribosome showing that would really alter the error rates. And so we know that a lot more than just simple Watson-Crick pairing is required to discriminate these correct versus incorrect tRNAs. And no one, even today, no one quite knows how this process works of the Trna selection. And one interesting aspect is that here in the pink, so here the black dot is an amino acid. If you look at one of these tRNAs, you have these 3 anticodon bases on the anticodon end, and then the body, like for example, the pink body connecting it. And then this light salmon color is supposed to represent one of these GTPases, elongation factor TU. And what happens is when a correct codon-anticodon pair is made and the ribosome acknowledges that, it actually sends a signal somehow to the other end near where the black dot is, and then a GTP hydrolysis event happens, and then the tRNA can slide all the way in. And so no one really knows exactly how that signaling works. No one knows quite how it picks out the correct from incorrect tRNAs. So there's still many mysteries even after almost half a century of work on this molecule. Okay, and then in terms of COVID, this talk is not about COVID, but the ribosome is important for COVID. And essentially, when the virus invades the host, it makes these mRNA-like molecules, and then it has to hijack the host ribosome somehow and get the host ribosomes to read out this pre-protein that's then processed into the spike protein and the other viral components. So there are a lot of COVID researchers out there looking at ribosomes right now. And then for the vaccine, as many of you may know, the Moderna and Pfizer vaccines are basically mRNA molecules inside a lipid nanoparticle. And then these mRNAs have to be read out by the ribosome. So it's important to know how the ribosome works, not only for antibiotics, but also for viral applications. Okay, so over the years, we've kind of put together this pipeline to try to make simulations that are as consistent as we can get them to experimental data. So one of the emphases we have is to really pay close attention to the latest structural data, the cryo-EM data that's coming out, and also the single molecule FRET data that's governing that's reporting on the dynamics of these ribosome complexes. So, so in our pipeline, we usually have an initial model, maybe one of the X-ray crystal structures or one of the cryo-EM structures of kind of a baseline state of the ribosome. And then we take cryo-EM reconstructions from those. And then we have a few different ways to come up with atomistic models that are highly consistent with those cryo-EM reconstructions. The latest one we have is working together with the Phoenix structural biology group, and we call this CryoFit or Phoenix.cryofit. And this, what this basically does is it morphs an already known all atom structure into a different functional state, in this case of the ribosome. And then once we have many different functional states, you know, there are at least 20 different functional states. So often we're just looking at transitions between 2 or 3 states for rate limiting processes. Once we have, say, 2 states, state A and state B, in the early days, and even nowadays, what we'll do is a kind of targeted or steered MD simulation to push the system from state A to state B. And this we found very useful to kind of assess out the problem and scope the problem to see how big the conformational changes are, where are the possible steric clashes, what the kind of gross overview of the conformational change looks like. And then we go in with all atom reduced model MD simulations where we can get very long timescales to look at spontaneous transitions back and forth between state A and state B, even calculate energy landscapes to look at a little bit of thermodynamics. And then finally, lately we've been putting in the actual FRET dyes into our simulations, and then we try to make very close comparisons between data measured in single molecule FRET labs with our MD simulations. Okay, so over the years, we've done a lot of different simulations. We started out back in 2000, and then we did our first fully atomistic simulation of the entire ribosome complex in 2002. And then we presented at a conference in 2004. And this was the first million atom simulation. Then we finally published it in 2005, where we were doing targeted MD simulations. And then we did some studies of drug binding to some of the drug binding sites on the ribosome using a technique called replica molecular dynamics simulations. And here we were just looking at, say, 1 to 10 microseconds of total sampling. And then after that, we moved on to doing longer timescale explicit solvent simulations of the full ribosome complex between 1 to 10 microseconds. And the latest things we've been doing are to go out to millisecond timescales, but using simplified force fields. So this is kind of a more esoteric technique, but it has some great advantages of having atomistic detail with good stereochemistry, but getting out to long timescales, and I'll talk a little bit more about that method soon. Okay, so first I'll talk about interpreting cryo-EM with MD simulation. And so there are a lot of different ways to come up with atomistic models for cryo-EM maps. And often when you've finally finished your 3D reconstruction, you do need to get some kind of atomistic model so people can look at it and interpret the data. And so there are two basic ways of doing the modeling. There's model building, which is kind of crystallography style, where you try to fit in every side chain into the EM map piece by piece. And then in cryo-EM, back in the days of low resolution cryo-EM, most people were trying to dock pieces of smaller pieces. So for example, the ribosome has 50 proteins. They might dock a few. Some of those proteins were solved by X-ray crystallography. Some fragments of RNA were solved by crystallography, and then people would dock those piece by piece into the cryo-EM map and then stitch them together. And then in the case of ribosomes, when the all atom X-ray structures came out, what we could do then is use MD simulation to try to sort of morph the full ribosome from one state into another state. And so for the model building, there are a lot of techniques. Phoenix has some, also Matt Baker's group in Baylor, Pathwalker, the Rosetta group, many different techniques to do model building. And then for morphing as well, Klaus Schulten, before Klaus Schulten was working on this, Florence Tama and Charlie Brooks and Joachim Frank were doing normal mode morphing. And so normal mode morphing or normal mode flexible fitting, they basically run normal modes, and then they try to get the sum of modes that will produce conformational change that can get the molecule into a state that fits into the cryo-EM map. And then real space refinement is one of the popular techniques to do more refinement conformational changes. And then MD flexible fit came out in 2007. That was with Klaus Schulten and Joachim Frank and Elizabeth Villa. And then we had an algorithm called MD fit in 2010 using this reduced model method that we can run on a desktop computer. And then more lately, there are many methods that came out since 2017. And ours is this cryo fit inside the Phoenix project. Okay, and one of our earliest attempts was back in 2002. And in this case, we were doing this old school way of just, this was Cheng Chung Tung, and I was working on this project as well at Los Alamos. And here, and as well as Simpson Joseph from San Diego. And here we basically had a motif library from the PDB back in 2002 when there wasn't a lot in the PDB for RNA. And we were able to stitch together a full 30 small cell unit of the ribosome. And then basically when we had two different conformations or choices to make, we would choose ones that fit better into the cryo-EM map. So this was the cryo-EM for one of Joachim Frank's maps for the small subunit of the E. coli ribosome. And then I went over most of these in the previous slides, but this is showing some of the references in case anyone wants to write those down. And then in our latest rendition of CryoFit, we've automated this process inside, linking it with Phoenix. And so this automatically installs the different scripts and packages you need. This current iteration is based on GROMACS. And so this will automatically install things and prepare the PDBs, generate the topologies, and run the simulations and report correlations to see how well you've done. And in our method, what we do is we do a MD simulation with a native MD, standard MD potential. And then, the second term is the standard MD potential. And then in the first term, we have this native contact term that I'll talk about in the next few slides. And this is this reduced simplified MD potential that's based on a given native state, for example, an X-ray crystallography structure of your system. And then finally, the third term is the cryo-EM term. And this gives a bias of the MD dynamics toward the cryo-EM. And so the native contact potential is very good for lower resolution maps, and then often we'll do turn up the high resolution part for to do the fine tuning of the maps. And then finally we have the EM term. And so the EM term is basically a correlation between the experimentally determined map and a simulated map. And then the simulated map is taken from the atomistic structure at a given time during the simulation. So this is updated throughout the simulation. And then in terms of what the native contact potential looks like, this is also called structure-based potential or all-atom go model. And in this case, the force field looks very much like a regular MD potential, but it's sort of hardwired toward your initial state, like the X-ray crystallography structure. But this is a lot different and more general than just simple harmonic potential locking things down to the crystal structure, because it allows for large scale conformational changes, huge conformational changes. But once a native contact forms, it tends to lock it in there. And so again, the parameters are taken from the crystallography structure, and we believe this is transferable because we have a server called the SMOG server where you can upload a PDB and then download your topology files to run in CryoFit, for example, or in just regular MD in any MD kernel you want to use. And so basically for any different PDB you give it, you can run an MD simulation. That's why we say it's transferable. Okay, and then so this is showing an example on the left-hand side of, we also do a lot of work on different non-coding RNA systems such as riboswitches. And this is showing an example of RNA folding. This is, the riboswitches are kind of for, segments of the messenger RNA, and they act to control gene expression by binding metabolites. And so this MD force field allows us to get full folding simulations that you couldn't really do with a regular explicit solvent, full-on explicit solvent simulations. And so one of the things we learned from this simulation is we could look at the order of helix formation. This happens to be the S-adenosyl methionine riboswitch, or the SAM1 riboswitch. And then we are looking at when do the 4 helices form in relation to ligand binding. And an analogy I like to draw is, if I can get that movie to play, let's see. I'm thinking I'm in some trouble. Maybe I'll exit out. Yeah, I think it's worth it exiting out. Get the movie to play. Okay, I think this was not in there properly. Okay, well, anyway, the idea of this is that once a native contact forms, then it tends to stick. And then once it's there, it can be disrupted by a large thermal fluctuation, but they tend to hold on to each other. And so we have native contacts and native dihedrals in the potential. Okay. Okay, so in terms of our CryoFit software, our main workflow is that first build the initial atomic structure. And then if the initial structure is quite different from the map, then we run the CryoFit package to do large scale conformational changes, and then refine, for example, with Phoenix real space refine. And if it's already very similar to the map, you can just go straight to real space refined. And what many people do is they build their model sidechain by sidechain already into the map, and then they run real space refine. But the advantage of CryoFit is that suppose you have the ribosome in one state and you want to get it in 5 or 6 other states, you can just run the morphing instead of having to rebuild your entire ribosome model again and get nice fits. Okay. Okay, so then what we encourage people do is to inspect the map and the model alignment at every step if you're running CryoFit. And then we also recommend performing successive fits. So we often will artificially smooth the cryo-EM map, say to a lower resolution, and then do kind of a lower resolution fit to the map. and then unsmooth it, or start with the original one and smooth at a higher resolution, and then refine more, and then finally go with the raw unfiltered map and then do the fine tuning there. And then again, we need to run real space refine to lock in the stereochemistry to really perfect it at the end. And so here, hopefully the movies play. Let's see. I think something's going on with my Zoom. Okay, I'll try exiting out of this. Okay, I think the movie, I'm having trouble with my movies. I'll just skip to this one. Hopefully this one works. Sorry. I just upgraded, so that's why I'm having trouble here. I'm just gonna, I apologize, I'm gonna stop sharing for a minute and to troubleshoot this. Sorry. Okay, I think all the other movies work except these three, so I think I'll be okay. Okay, so I apologize. Yeah, I'm learning the lesson never upgrade before a presentation. So, okay, so anyway, these were just some examples showing the fits here, but I'll have to skip over those. Okay, so some of the other work we've been doing is on calibrating FRET simulations with single molecule FRET data. And so here we've been working closely with Scott Blanchard's group at who's now at St. Jude's in Tennessee. And he's looking at, just as a test system, this leucine, isoleucine valine binding protein. And what we were trying to do here was try to get apples to apples comparisons between all atom simulations and some single molecule FRET data. And so in this system, the LIV-BP system, he's seeing a open state and a closed state, a clamshell-like conformational change, where the open state, here we're showing where fluorescent labels are, donor and acceptors here, and the open state gives a lower FRET value, and then the shut state gives a higher FRET value. And then, so here's just some examples of some of his histograms that he's seeing. And so here, you can see the lower FRET state and the higher FRET state, and he'll often see excursions between the two. And so one of the things we wanted to do was simulate this and try to make contact with his single molecule FRET data. And what he was doing was pushing toward higher time resolution. And so here, he was looking at quite low time resolution, 100 millisecond, and then 15 down to 1 millisecond. Then he actually has some trajectories with quarter millisecond time resolution. And so then we went in with our reduced model simulations of this protein system to try to see if we could get results that agreed with his experiments. And so we were using he also makes different kinds of FRET dyes to improve the time resolution. So here he has these newer FRET dyes that give cleaner signal to noise and better time resolution. So we modeled those into our LIV-BP system, and we did these structure-based simulations to model the conformational changes here. And then in this case, we have two different native states. an open native state and a closed native state, and then we're looking at transitions between the two. And so for individual trajectories, we smooth the trajectories to try to mimic the time resolution he's seeing, and then we ran quite far out in our trajectories as well, so that the units here are in seconds. And so we managed to do some 10-second long simulations, again with the reduced force field, but with all the atoms. And so we can see similar fluctuation behavior for the top panels are 100 millisecond time resolution. The bottom panels are 1/4 millisecond time resolution. And then we looked at histograms as a function of time. And in the color heat maps, we can see we get pretty good agreement. The top here is the experiment, and the bottom is the simulation. And what we see in the experiments is that when we go to higher, when we are at very low time resolution, you don't even resolve this open closed motion. But when you go to very high time resolution, you can resolve individual open close events. And so we see the same thing in our reduced model description molecular simulations. And then finally, we see at the bottom just averaging over the full trajectories, the histograms. And then as you go to higher and higher time resolution, you can resolve the peaks. Okay, so now I'll talk a little bit about explicit solvent simulations of a single basin. And so for RNA systems in general, we encourage people to use a very careful equilibration protocol. And we do, because the ions, especially magnesium, are so important, we encourage good sampling of the ions. And so here, what we do is basically a run without water, but a fixed solute, in our case, the fixed ribosome. And then we use a larger radius of the ions to mimic hydration shells around magnesium and the hydration of the potassium and chloride. And then in our own special protocol is to then solvate that system with SBCE water, again, equilibrating the water with fixed ions and the fixed RNA. And then we release the ions and we release the RNA and slowly heating up things. And we try to do a lot of equilibration before we go into production mode. And when we do this, we can still use one of the older RNA force fields, but get very stable trajectories over microseconds. And so this is looking at some of our equilibration steps, showing on the left the magnesium condensing onto the RNA as you're equilibrating without water here, but a big radius to imitate the water, and then the chloride kind of blowing off the RNA. And then we choose our numbers of ions so that when we're far away from the ribosome, we have physiological concentrations. Okay, and then this is showing a picture of the ribosome solvated with water. And then here, we can look at the water density around the ribosome. And then as we zoom in, we can look at water coordination around the RNA in the grooves of the RNA helices and so forth. And then, and we can also look at the one area of interest in ribosome research is that we have these two subunits, the small subunit and the large subunit. How do they interface together? It's kind of an interesting physics problem because somehow they have to be glued together, but they also have to have viscosity, or they have to be slippery, so they can slide over each other to do this rotational motion. So somehow they're glued together so they don't fall apart, but yet they can easily slide over each other when they need to. And we think this is mediated by bound water and bound ions. And then this is just looking at one of the tRNAs inside the ribosome and where the water is. Okay, then this is showing a explicit solvent simulation of the 70S complex. And here, if you look in the lower left, we're looking at kind of different, each frame is representing different time scales. So we're increasing the rate of view by every 10x now. And then these simulations were done inside a box of water with ions, but then we're gonna turn off the ions and water so we can see the ribosome moving around. And then you can see that our protocol gives very solid RNA trajectories. And we can see all the double-stranded RNA helices are pretty much rock solid. And then this region here, basically the regions that are supposed to be flexible are highly flexible. So we were just looking at this L7, L12 stalk that's this arm that's always swaying wildly about in solution, recruiting the G proteins. And then here we're looking at the small subunit in blue. And then as we zoom in, we can look at the tRNA in yellow, and then the codon-anticodon interaction happening between yellow and green. And we see really nice stable RNA helices and RNA base pairs. And then as we zoom out, we can look at the full complex and see where the tRNAs sit here in yellow and red, where they sit in the ribosome. And then finally, we can see the complex. So basically the core is quite stable, and then the two arms are fluctuating as they should be. Okay, and so in this study, we did a lot of trajectories, which added up. We had one microsecond trajectory, and then all the others added up to about 3 microseconds. And we used this to try to look at, to try to see if we could extract useful information from these fairly short time scale, only a few microsecond simulations of the ribosome. And so in one study, what we were doing was trying to estimate, basically to look at the diffusion of the ribosome inside a single basin and use that to get more realistic estimates of prefactors for transition rates. And so in this study, we were basically saying, if we already measure the rate experimentally from single molecule FET or rapid kinetics or whatnot, can we then somehow, knowing that rate, get a better estimate of the energy barrier heights? And so what we did is, with our MD simulation in a single basin, we looked at the diffusion rates for different order parameters and came up with estimates for the delta G's corresponding to already known transition rates. And we believe these are more accurate than typical textbook estimates. And then we came up with these tables to go from the experimental rates on the vertical axis to barrier heights on the horizontal axis. Okay, so now I'll show some simulations of the ribosome using targeted molecular dynamics. And so here we're simulating single barrier crossing events using targeted MD. And these events, these studies are useful because even though they're quite short time scale, they enable you to see the kind of sterics of the problem of tRNA moving through the ribosome. And in our case, we're looking at an event called accommodation, where the tRNA, once GTP hydrolysis happens, and after the tRNA is recruited to the ribosome, then it moves all the way into the ribosome. This is called accommodation of the tRNA, and it's an important event that happens during Trna selection. And so here we were running targeted MD using Lagrange multiplier constraints on the RMS to target at each time step, and then we decreased that constraint as a function of time. And then one point to make here is that these kinds of transitions are relatively straightforward, and that's why targeted MD works. And basically the ribosome is kind of, or I'm sorry, the transfer RNA here shown in blue is kind of jam-packed into the ribosome, and then it has to kind of squirm through this cavern. And there's not a whole lot of things it can do. You can kind of see the trajectory in your head if you look at the ribosome. There's no way it can unfold. The helices can't really untwist. It can't go upside down or really flex outward. There's a very narrow cavern that it has to go through, and that's why targeted MD works pretty well in this system. Okay, so here we're showing one of these trajectories, and then in green is the messenger RNA, in purple is the small subunit, and yellow is this tRNA that's accommodating into the ribosome. And we were able to use this study, this we did back in 2005, we published it, and we were able to look sterically at where the tRNA interacts with the ribosome during this transition. So again, you could kind of see the transition in your head, but here we could get the specifics of where the tRNA needs to pass by on the large subunit on the top half here during the accommodation transition. And so we were able to point out a lot of different ribosomal RNA nucleotides that were very important for this transition. So those are highlighted here in pink and purple and so forth as the tRNA moves in. And most of these are actually universally conserved. So in the ribosome, there are these things called universally conserved nucleotides that are actually identical in every species that's ever been sequenced. And then we hypothesized that these three might be really important for this process. And then as we look here, we can see these universally conserved nucleotides. And then finally, at the end, we can get a closer look of the tRNA moving in. And the end, this tail end of the tRNA is called the 3' CCA end, and the green is the amino acid. And then we named this helix the accommodation corridor, where it skirts through at the very end. And it turns out that that this accommodation corridor and also helix 89 are, we predicted they would be important antibiotic targets, and people are looking at them now for leads. In particular, vernomycin binds to helix 89. And then one of the nice advantages of targeted MD is that you can basically dial in the beginning and end states, and you're assured that you'll get there. Okay, so we made these different predictions, and several groups validated these. John Didman's lab at Maryland, and also the Donsova group showed that our three, well, many of the predictions were correct, but importantly, this mutation of 2492 to 2493 is actually lethal, so it's extremely important. Okay, so now I'm gonna talk about the last kind of simulation, which are these free simulations of transitions using the same reduced MD model that we used for the cryo-fit in the beginning of the talk. And so here, you know, we've seen the ribosome field evolve a lot in the last 20 years. And back in 2000, people were thinking that it kind of does this lockstep robot style movement through all the sub-steps of protein synthesis. And nowadays, people are thinking of this more about more in terms of energy landscapes, where you may have two states and the system's fluctuating. Say it's sitting in state A, and then there's a metastable state nearby, state B, and it fluctuates back and forth. And then when an event happens, it triggers a remodeling of the energy landscape to shift the equilibrium along in this process of protein synthesis. So in terms of Trna selection, what we did is we went in with our native state models, or these reduced description models, and we did the same kind of accommodation simulation, but now allowing spontaneous fluctuations. And so here we could look at the tRNA kind of go in and out and in and out, and then eventually make it all the way to the end state. And so here we're seeing a more zoomed in view of the yellow tRNA trying to move in. And then one thing we identified in our first study and in this study was that helix 89 we thought was going to be very important. And the story that emerged from this study was that it's positioned to contact the tRNA during the accommodation, but basically the tRNA is trying to get in, and then when helix 89 has a favorable fluctuation, then the yellow tRNA can scoot by and make it into its accommodated state. And so here we're looking at this process and how that CCA can go in. And then here we can even zoom in more and see this 3' CCA end sampling around and then making it to the final accommodated state. Just let this play out. Yes, okay. Okay, and so then we were comparing with, this was done over 10 years ago, so this was comparing with Scott Blanchard's data in single molecule FRET, and this is before we were going out to really long time scales, but here just qualitatively, we would see these reversible excursions going in and out. And then in the stuff I showed you on the one protein in the beginning of the talk there, we're actually doing apples to apples comparisons in actual terms of time scale. And we're right now in the process of porting that to full ribosome simulations. And then on this older ribosome study we did, we were doing kind of cross-validation where we were comparing the reduced description models to the explicit solvent models. And so on the left-hand side, the reduced description actually gives us the heat map. So there we can sample hundreds or even thousands of these transitions. And then with explicit solvent, we're only allowed to sample one basin basically, and not the transitions. But we say it's cross-validation because the reduced model actually can validate the explicit solvent in terms of how much sampling do you need. And then on the right-hand side, we were comparing targeted simulations in explicit solvent in the single traces to the heat maps in the reduced models. But here it gives you some validation of the reduced description transitions. Okay, and then some of the latest stuff we're doing with this is we're looking at cognate versus near cognate systems. So I talked a little bit in the beginning about how the ribosome has to reject incorrect tRNAs. And so what we're trying to do now is to look at correct versus incorrect tRNAs. And this is showing an energy landscape with different order parameters or parameters describing the process, where along the x-axis is the codon to anticodon distance. The x-axis is basically the progress of the elbow of the tRNA moving all the way in. And we can see for the correct cognate case, on the left-hand side, we have one kind of pathway. And then in the incorrect case on the right-hand side, or the near cognate case, we have two different pathways that the tRNA can take. And so we think here that the tRNA tends to get hung up going through here. Okay, so lastly, I'm kind of running out of time, but I'll talk a little bit about translocation, which is far more complicated than the accommodation step. And in translocation, there are many, many different sub-steps and lots of different conformational changes of the whole ribosome. And this is kind of the holy grail of bacterial ribosome research to understand translocation. So our strategy was we started with accommodation, which was simpler. It really only involves the tRNA, and then to move on to translocation, which we've also been working on since like 2003 or 2004. But even the experimentalists don't have a clear idea of what the different states are during translocation. So we kind of work together with cryo-EM groups. We're doing some of our own cryo-EM now into a single molecule to try to get a very clear, precise picture of what happens during translocation. This is the movement of the tRNA all the way through the ribosome. Okay, good. This movie's working. So I talked a little bit before about the energy landscape view of protein synthesis. And here the idea is that at any given time, the system may be in one state, but it's next to a metastable state. And so it's kind of sloshing around, but transiently sampling this neighboring state. And then the idea is when an event happens, like the chemistry, the peptide bond formation, or G factor binding, that it remodels the energy landscape to move the system forward in this progression. And in this way, it's thought we can get those tRNAs all the way from coming into the ribosome to state A, to state P, to state E, and then out, which given the ribosome is quite big, this is kind of a huge amount of gymnastics that has to happen with both the tRNA and the ribosome. Okay, so there are lots of different conformational changes that the ribosome can do. The most famous is this, they used to call this ratchet-like motion, where you have the two subunits rotating with respect to each other. They also call it 30S rotation. 30S is a small subunit. And then there's also head swivel, which is a newer one, but we think is even more important than the rotation, because we believe, remember you have the body and the head of the small subunit, the mRNA is wrapped around the neck. And we believe the head is kind of really the thing moving the mRNA and the tRNAs through the ribosome. And then this is just showing, putting some of the steps together. First EFG binding, and then the ratchet-like motion, or the 30S rotation, then the head swivel, and then everything unswivels and unrotates. And in this way, the tRNAs are shuttled through the ribosome during translocation. And so this is let's see if this is moving to work. Yes, okay. So then this is showing an older set of simulations we did using reduced models and looking at, this was combining reduced model simulation with targeted MD, just to give an overview of what these conformational changes look like. And so this is the ratchet motion, or the whole 30S body rotating. And that's coincident with this pink tRNA diffusing over to this so-called hybrid state. And then something happens with EFG and the head swivels. And you can see, if you look carefully, that looking down on the head, it pivoting. And then this is chopping off the head. And you can see that the messenger RNA, shown in gray, does most of its movement during this head swivel step. And then finally, things kind of reset where the head unswivels and the body unrotates, and we get to the final state. Okay. And so one of the first studies we did was to look at that initial movement happening during the ratchet motion, where the red tRNA moves to this hybrid state. This is called hybrid state formation. And we did a similar analysis with this problem as we did for accommodation, where we looked at the transitions between the two states to look at the landscapes. And then also we identified these universally conserved bases that are kind of this corridor for hybrid state formation. And then the latest thing we're doing on this is to look at the head swivel motion. And our colleague at St. Jude's, Blanchard, discovered a new state in single molecule. And we're going in now with structural modeling to model that state, combining cryo-EM and single molecule FRET. And so we basically had the nomenclature is kind of esoteric here, but we have, and there's many different kinds of brands of nomenclature, but basically there's a before head swivel and after head swivel, pre and post, that we're looking at. And so we were trying to model those, and also this newer state that our colleague discovered. So here, the newer state was shown here, where he was looking at FRET dyes to specifically measure the head swivel motion. So the head is shown here on the left in light blue of the small subunit. He has a dye in green attached to that. And then the large subunit is the upper half, and one protein on the large subunit is shown in yellow. He has a red dye attached to that. And that way you can monitor the motion of the head swiveling with respect to the large subunit. And then he was able to show that basically you see this dip go way down in his FRET data. And this he was hypothesizing is a new state in 3 that's rate limiting to the whole process of translocation. And so we went in and tried to model that. So we put in his FRET dyes at the right places. And then here we're showing just sampling of the FRET dye, the cloud of sampling of the FRET dye inside the ribosome. In this case, the red cloud is the tRNA FRET dye, the yellow is, or the green is, in this case, another tRNA FRET dye. And then also, or sorry, the yellow is the other FRET dye on the tRNA, and the green is a FRET dye on this elongation factor G. And then we also have the FRET dyes on the head and the large subunit as well. And so that's shown here. If you look carefully, you can see some of the FRET dyes. Let's see, this was another movie that didn't work. Okay, but so then here we can see in the schematic of things where this new state fits, basically the head swivels, and then he was hypothesizing that something called super swivel, where it goes even further than the normal head swivel and then back. And then this is a lot of nitty-gritty of ribosomology here, but we were able to do our simulations and get FRET dye distances pretty consistent with the measured distances for these studies, especially for INT3, where we had 63 angstroms and he was estimating 65 angstroms. And so we think we have a pretty good model for this super swivel. And then we came up with a story on how that when the head shown here in pink is moving to its first swiveled state and then the super swiveled state, we think this gives an opportunity for the E-site tRNA in turquoise to dissociate or disengage and then allow reverse swivel to happen to resolve this huge steric clash. Okay, and then in this study, we've identified many universally conserved regions that are important for this process. And we put this in the context of a lot of the ribosome structures that have been done, and we put those as scatter dots on these plots to kind of see where our model sits in the context of this. just about out of time, so I'm going to skip through this, but we've looked very closely at these transitions and see that the universally conserved regions on the head and body are important for this. Yeah, and then also basically the way we did the model is we did the head swivel, and then we continued it out further until we hit steric clashes from other ribosomal proteins. And it happened to be that when the farthest you can go out is 22 degrees, and that also 22 degrees directly corresponds to moving the tRNA and the mRNA exactly to their fully translocated positions. So yeah, and then we have another story about the E site dissociating, but I'll skip over that. Okay. Okay, so so I'll just wrap up and say that we came up with this cryo-fit technique that produces atomistic models highly consistent with cryo-EM data. The structure-based simulations allow long time scale spontaneous transitions while preserving stereochemistry. And then this new super swivel model we have is consistent with the single molecule FRET data, and it delivers the tRNA and mRNA to their final translocated positions. And then I'd like to thank the people who did the work here, especially Wataru Nishima and Dylan Girodot, and all of our ribosome collaborators, especially Scott Blanchard. And thank you for your attention.
Okay, thanks very much, Karissa. We don't have a good way of applauding on Zoom, but I really enjoyed it. And we kind of just do a quick free-for-all for a few questions, and just unmute yourself if you want to ask a question. I'll start with one about magnesium versus potassium ions. And one of your slides, and I should know these things more, you had 7 millimolar magnesium and 100 millimolar potassium. So my first question is do we know that where those numbers come from? And more importantly, have you looked at how much of the magnesium actually ends up in contact with neutralizing the RNA versus the monovalent ion, given those bulk concentrations?
Yeah, that's a great question. And nowadays we tend to do 2 millimolar. I think this was controversial in the ribosome field over what is physiological magnesium concentration, and people shifted more to 2 millimolar magnesium in the last 10 years, I'd say. So we shifted along with them. I'm not an expert at how they measure that in cells, so I can't really comment on that. I just kind of go with what they tell me. And then in terms of what happens in the simulations, we've been doing a lot of detailed simulations of the concentrations in smaller RNA systems like the ribose switches. And so there, we're always trying to improve our protocols. So what we do nowadays is basically we do an iterative process where we put in a bunch of magnesium, and then we see it condensing, and then we make measurements of the concentration away from the solute, and we try to match that with the physiological concentration. Then in addition, David Draper has and others have a measurement they call the preferential interaction coefficient, which you are well aware of. And so we also try to make contact with those data as well. And so we see it varies like from ribose switch to ribose switch in terms of the fraction of magnesium versus the potassium condensing, I guess. And so so we just try to do the best we can making contact with those preferential interaction coefficients, I guess. But I don't have a good answer to your question.
Well, no, it is a good answer. Thank you.